Okay, moving on to section 8, we're going to talk a little bit about the respiratory centers. These respiratory centers are located within the brainstem itself, and the respiratory centers are responsible for controlling our breathing. We don't know a whole lot about all the respiratory centers or a lot of detail about them, so it's kind of just a general overview is what we're going to go through today. But regardless, all of our respiratory centers that we have are receiving input from a variety of sources like our chemoreceptors and higher brain centers that are going to help influence the rate of our respiration. So our two main respiratory centers reside in the medulla oblongata, and that's our ventral respiratory group, or VRG, and our dorsal respiratory group, or DRG. If we're talking about the ventral respiratory group, it's called that because of its location. So it extends from the ventral portion of the brainstem to the pons and medulla area. Our dorsal respiratory group, or DRG, that's going to be closer to the dorsal end, very close to cranial nerve number 9. So for the ventral respiratory group, this is what is thought, again, not a lot is known, but what is believed to be the rhythm generating center of our respiratory rate is thought to be the VRG, and it just contains a cluster of neurons that are going to fire while we're going through inspiration, and then we also have clusters of neurons that are going to fire if we're going through the process of expiration. So it's kind of like this dance between neurons firing for inspiration and neurons firing for expiration. So it's referred to as mutual inhibition. And again, this is going to set our normal respiratory rate, or thought to set our normal respiratory rate and rhythm, which is approximately 12 to 16 breaths per minute. When we're talking about the inspiratory neurons and the expiratory neurons, they're going to activate different things. So our inspiratory neurons are going to excite the inspiratory muscles, so our diaphragm and external intercostals, and then our expiratory neurons are going to inhibit the firing of the inspiratory neuron. The dorsal respiratory group is thought to act as an inspiratory center, possibly performing a lot of the same tasks as our VRG, but right now they really don't understand too much about this. There's not a whole lot, but they think that it's going to send information again to the VRG, but not a whole lot is known. The third center is our pontine respiratory center. And again, our VRG is going to be the one that actually generates our basic respiratory rhythm, but it's thought that the pontine respiratory center can send information to help influence and modify the activity of the neurons in the VRG. So they can act to basically smooth out the transition between our inspiration and expiration process. And they can also help transmit information during our speech or vocalization, also while we're sleeping and during exercise, so that we can alter our basic respiratory rhythm. Why do they think that the pontine respiratory center is responsible for helping influence the rate? Well, if we do some sort of lesion in this area in our brain, the patient ends up with prolonged inspirations, meaning it can't alter its basic rhythm. It's kind of prolonged, and that's the new basic respiratory rhythm. So they believe that it influences it because if it's damaged, we can't really control the rhythm rate anymore. Okay, so again, this section's kind of just brief because they really don't know a lot about how the respiratory rhythm is generated, or they can't really explain how we initiate it. There is one hypothesis that there are pacemaker neurons, and these pacemaker neurons have intrinsic ability to go in and fire, just like the pacemaker cells that we talked about in our heart. The pacemaker-like activity has been shown in some VRG neurons, but not all of them. And if we were to suppress these VRG neurons, their activity However, it doesn't get rid of our breathing at all. So again, that's just one hypothesis, that there's pacemaker neurons, just like we saw in the pacemaker cells of our heart. But it does lead us to think that there is a more widely accepted hypothesis that our normal respiratory rhythm actually results from that reciprocal inhibition that I was talking about earlier between those interconnected neurons in the medulla. Rather than having a single set of pacemaker neurons, there's actually more likely to be two or even more sets that can go in and inhibit each other and then cycle their activity to help generate the rhythm itself. Okay, factors influencing breathing rate and depth. Inspiration depth, when we're talking about that, is determined by how active are the respiratory centers and their ability to stimulate the motor neurons that are actually serving the respiratory muscles. So the greater the stimulation, the greater the number of motor units that are recruited, and the greater the number of motor units that are recruited or excited means the greater force the respiratory muscle is going to be able to undergo for contraction. So respiratory rate is determined by how long the inspiratory center is active and then how quickly it is also turned off. So if we change any of our body demands, so say we're sleeping versus exercising, we can go in and modify the depth and the rate of our breathing. And those respiratory centers that we talked about previously, the ones in the medulla and the pons, those are sensitive to both the excitatory and inhibitory stimuli. Okay, so when we're talking about what they can be affected by, there's 4 different things that can influence our breathing rate and depth. One is chemical factors. The second one is our higher brain centers. We can also have pulmonary irritant reflexes that can come into play, and inflation reflex as well. So let's talk about chemical factors first. The most important chemical factors that affect our breathing rate are going to be the partial pressure of carbon dioxide, the partial pressure of oxygen, as well as the pH or the hydrogen ion concentration in our arterial blood. So we talked about these before when we're talking about the cardiovascular system, and that if we have increases in carbon dioxide, what that happens, or if we don't have enough oxygen, or if we have too low of a pH and we become too acidic. Well, the chemical factors, how do we know what levels these different chemicals are at? Well, we have some chemoreceptors, and the chemoreceptors are found in 2 major body locations. So we have central chemoreceptors and then peripheral chemoreceptors. The central chemoreceptors are central, so they're located throughout the brainstem. If we're talking about peripheral chemoreceptors, those are going to be found in our carotid arteries and our aortic arch. So let's talk about each one of these. If we're talking about carbon dioxide or the partial pressure of carbon dioxide is going to be the absolute most powerful and most closely controlled concentration of a chemical that is going to influence our actual respiratory rate. So normally, if we're talking about the partial pressure of carbon dioxide in our arteries, that's about 40 millimeters of mercury. And we talked about this. You don't have to know specific numbers or anything. But we try to maintain this 40 millimeters of mercury within 3 millimeter of mercury units. So we don't want it to go above 43 millimeters of mercury or below 37 millimeters of mercury in order for our body to maintain homeostasis. So in the event that our partial pressure carbon dioxide levels rise in our blood, this condition, if we have elevated carbon dioxide, is called hypercapnia. What happens is we have carbon dioxide that's going to begin accumulating in the brain, but it's not going to be accumulating as carbon dioxide. It's actually going to be in its carbonic acid form. So we talked about how we transfer or transport carbon dioxide from one portion of our body to another, and it's actually by going into our erythrocyte and being converted into carbonic acid, which can then be converted into the bicarbonate ion. But when we start to accumulate carbon dioxide in our brain, that's going to be in the form of carbonic acid. When we have carbonic acid, the hydrogen that's attached to this, which makes it an acid, actually is going to dissociate from the carbonic acid compound itself. Once it releases the hydrogen ion, that means the hydrogen ion concentration is going to increase. The higher the hydrogen ion concentration you have, that means the lower the pH or the more acidic that you have. So again, this is the same reaction that occurs when we had carbon dioxide entering into the erythrocyte. So once we start to build that supply or that concentration of our hydrogen ion, that's going to excite our central chemoreceptors that we talked about earlier that were located in our brain stem. And what's going to happen is once these excite those central chemoreceptors, the depth and the rate of our breathing is going to increase. And the point of it increasing is so that we can increase our alveolar ventilation and quickly flush that carbon dioxide out of our blood. And again, losing the carbon dioxide because it's an acid is going to help raise our pH or make it more basic. So the reverse is true if we're talking about low levels of carbon dioxide. Respiration is going to become slow and shallow. You can get something called apnea, where your breathing when you stop breathing or you have breathing cessation, when your levels of carbon dioxide drop abnormally low. So a lot of times swimmers, they would actually hyperventilate to let them hold their breath longer. If you start to hyperventilate, it causes a drop in the partial pressure of your carbon dioxide because you're breathing out much faster than you're bringing oxygen in, and it causes a delay in respiration. So if you hyperventilate, it's going to be longer before you actually want to take a breath, and they can hold their breath longer for the swimming. It can be really, really dangerous, of course. People and swimmers who have practiced this before have actually passed out, and it could be potentially fatal if you're in the water as well. So this is just a flow chart telling you what happens in the event that your partial pressure carbon dioxide gets too high, and what centers is going to activate to ultimately end up restoring our homeostatic level of carbon dioxide. Okay, influence of the partial pressure of oxygen. This is going to be the responsibility of our peripheral chemoreceptors, which are found in our aortic arch, and again, those carotid arteries. And in the peripheral chemoreceptors, there are special cells that are sensitive to the concentration of oxygen in these areas. The major oxygen centers are actually in the carotid bodies, not the aortic arch itself. So if we're talking about normal conditions, kind of like at rest, and we have declining partial pressure of oxygen, that only has a slight effect on ventilation itself, if we're talking about that. But if we're talking about huge amounts of partial pressure of oxygen being lowered, then that's going to require a substantial drop in arterial partial pressure of oxygen below 60 millimeters of mercury before our oxygen levels can end up becoming a major stimulus for increased ventilation. Because remember, the levels of carbon dioxide is going to be our main contributor to our respiratory rate, not oxygen. So we have to have pretty substantial drops or changes in our oxygen levels in order for oxygen to be a major stimulus for ventilation. Okay, arterial pH. So if we have any changes in pH of our arteries, that can also modify our respiratory rate and rhythm, even if we have normal levels of carbon dioxide and oxygen. The reason why is because the hydrogen ion cannot cross our blood-brain barrier. So the increased ventilation that occurs is going to actually be in response to the pH in our arteries and mediated through those peripheral chemoreceptors itself. Decreased pH can also reflect us retaining a lot of carbon dioxide, but it could also result from metabolic causes. So things like accumulation of lactic acid if we're exercising and our skeletal muscle isn't getting enough oxygen, so it has to go through lactic acid fermentation, or as well as fatty acid metabolites or ketone bodies that are also accumulating or being built up. So the respiratory system can basically controls attempt to raise our pH by increasing our respiratory rate and our depth. Okay, summary of chemical factors. Again, this is probably a lot. Just in general, which one of the three we talked about, the partial pressure of carbon dioxide, the partial pressure of oxygen, and the pH in our arteries. Which of these is the most powerful respiratory stimulant? Which one's the least powerful respiratory stimulant? How does it work? Is it central chemoreceptors, peripheral chemoreceptors that are sensing these different things? So again, if we're talking about carbon dioxide, if we have elevated carbon dioxide levels, that is the absolute most powerful respiratory stimulant. We're going to have carbon dioxide in carbonic acid form. The carbonic acid is going to release its hydrogen ion, which means we're going to have an increase in hydrogen ion concentration or a lower pH and be more acidic. That's going to cause us to increase our respiratory rate and depth. If we have too low levels of carbon dioxide, that's going to completely depress our respiration overall. If we're talking about oxygen, it does affect our breathing, but kind of indirectly, and those are going to be sensed, if we're talking about oxygen, by our peripheral chemoreceptors. If we have low partial pressure of oxygen, that's going to augment our partial pressure of carbon dioxide. And if we have high levels of oxygen, that can diminish completely the effectiveness of the carbon dioxide stimulation. And then if we're talking about arterial partial pressure, arterial pH, that's going to result from carbon dioxide retention. We have too much in our blood, or metabolic factors that we talked about, like lactic acid fermentation, or even indirectly through peripheral chemoreceptors. Okay, higher brain centers. If we're talking about higher brain centers, we're really focused on the hypothalamus. So the hypothalamic controls act through the limbic system. So we're talking about strong emotions, pain. Those can all send signals to the respiratory center and modify the respiratory rate. Cortical controls, we're talking about direct signals going from the cerebral cortex, and they're going to completely bypass those medullary controls, the VRG and the DRG that we were talking about. So an example is if we voluntarily hold our breath, at least until the brainstem reinstates the breathing. Basically, it forces us to breathe. That's why if you've ever taken note of a drowned victim, they always have water in their lungs, because at some point in time, your body forces you to breathe, and if you're underwater, you take in all the water that you're in. Okay, pulmonary irritant reflexes. Again, these are other things 2, 3, and 4 that can affect and modify our respiratory rate. Just in general, how these work. But more specifically, the ones you should really focus on are carbon dioxide, arterial pH, and oxygen levels. And then these recognize that they also can affect our respiratory rate. So if we're talking about pulmonary irritant reflexes, our lungs, they also contain receptors that can respond to a variety of different types of irritants. When these receptors become activated, they can communicate with respiratory centers, and different things can happen. So we can promote reflexive constriction of our air passages. We can cause us to cough or sneeze or some other sort of reflex, depending on where the irritant is. And the last one is going to be basically the inflation reflex, the Herring-Brewer reflex, Breuer reflex. This is where we're talking about receptors that are in our pleura and the airways that are stimulated by lung inflation. And these receptors are going to signal the respiratory centers in our medulla, sending inhibitory impulses that basically shut down inspiration and allow expiration to occur. And they may act as a protective response more than as part of our normal regulatory respiratory mechanism.